Thank you very much to all the organizers, and especially to Victor Acuña, who very kindly contacted me, and he told me that the topic of the meeting was mainly humans, which was also highlighted in the cloud. I usually don't work with humans, so today I'm presenting you the little work we do with human genomics. So we're doing this to try and understand and characterize better the signatures associated with longevity, both at the cellular level, and then the second part of the talk will briefly touch on some of the work being done on a comparative analysis using mammals. So in my group, what we are interested in is trying to link the genotype to the phenotype. And I think that's a general topic across labs doing genomics. So now we have extensive amounts of data for genomic and transcriptomics, and increasingly for proteomics as well. But we still don't understand fully the language of genes, so how genes and genomes are creating or leading to phenotype. So as I said, I usually don't work with humans. Instead, we basically tend to work with the favorite organisms of nice people I meet in random circumstances, and I get excited about their model organism or their taxonomic group of interest, and we start working with them. So I've been very lucky to have worked in the last 4 years with lots of people. Some of them are there, and they tend to work with plants or fungi or all sorts of animals. I haven't yet worked on bacteria, but that might change at some point. So we use mutant models to try and understand the molecular basis of complex phenotypes. We use single species analysis and also comparative analysis. And we tend to use, I like mostly transcriptomes, but we also do lots of molecular evolution kind of studies and other genomic analyses as well. So today we're talking about aging. Aging is very important. It's actually the main risk factor for death, of course. But it's also, well, we hear about obesity, right? We're scared about eating biscuits or eating, there weren't any biscuits outside probably precisely for that. We talk a lot about smoking, and that's bad for you, you'll die early. But actually, the biggest risk factor for dying of or acquiring any of these chronic illnesses or other diseases that might lead to death is how old you are, right? So we don't really understand how the aging process works. There are lots of different cellular processes and overall body processes that fail over time and collectively lead to a higher incidence of disease or just generalized organ failure, and you end up dying. So what we were interested in is a process of how cells are maintained after they stop dividing. So when we have a fully differentiated cell that stops dividing, then it has to stay alive while not undergoing mitosis. So the best example is our neurons. They have to stay alive after we're born, and there's hardly any new neurons being added to our brains after we're born. So they need to stay alive, they need to stay healthy, and that's actually a very complex process we don't fully understand. So what the analysis I'm going to show you now are aimed to try and find out whether there is a set of a genomic signature associated with increased longevity. Some people, when we started working on this, told me you're not going to find anything because every single cell type in the body has its own mechanism to maintain post-mitotic cell longevity. So we still tried despite that. This work was done with Atahualpa Castillo and Jimena Monzon. Both of them are from the undergrad program of genomic sciences. They did their PhD with me and also with Humberto Gutierrez, who's currently at the University of Lincoln. We were working with a very limited set of transcriptome data from different tissues, so we were using only 7. And for these, we had estimates of the postmitotic longevity of cells that were calculated using radioactive signals that were stored or acquired by human cells during nuclear tests that were done in the 1950s. The numbers here are just the numbers of years that each of these cell types live after they stop dividing. So what we did was something very simple. We made a correlation for the expression level and the longevity for the 7 tissues, and the way we did it was we removed each time one of the 7 tissues, so the correlations were actually made on 6, and then we obtained the average. So this was just to try and avoid that the brain that actually lives, brain cells that live an inordinate amount of time would bias the whole analysis. So with that, we selected a set of 81 genes that were correlated with longevity. So not surprisingly, of course, if you take the average expression level of all those 81 genes and you put them together against longevity, then what you see is that with increased longevity, the mean expression of those 81 genes goes up. And this is because we selected them precisely because they were correlating with longevity. So this is just to confirm that indeed we found that signal. So the next thing we did was to try and see whether we could find a signal for them to be acting in concert. Are they participating in as a group in regulating postmitotic cell maintenance? So for that, we used an approach of calculating co-expression levels. If you find a group of genes that are working together, then what you expect is that when you obtain the correlations of their expression level over much larger number of different tissues, they should be having their expression levels across 28 tissues we sampled to be highly correlated. And so the network should look something like that. So if you have a group of genes and they are interacting with each other, then they should show a pattern of high co-expression among them. In the opposite case, if the genes are not really interacting with each other, what you expect is that when you do the correlations, you hardly find any connections linking them. Yeah, so that means that each of those 81 genes would be participating with other groups of genes rather than acting in concert. So that's the thing we were testing. So this here is mean co-expression for the group of genes. So this is the 81 group of genes, and these are randomly selected ones. So what we find is that when we look at the 4 most long-lived tissues from our data set of only 7, remember, then what we find is that the level of co-expression of those genes among that set is very high compared to randomly selected genes. When we look at the short-lived tissues, then we find that there is no difference between the real 81 set of genes against the randomly selected samples. We also did it with additional data, so there was a different set of transcriptome data that was not used in the original analysis for brain, muscle, and skin. And what we find is that the degree of co-expression of those 81 genes across different brain areas is much higher than that expected by chance, selecting random sets of genes in the same tissues, or that found in other non-so longeve tissues such as muscle or skin. So then what we did was to try and test whether the sets of genes that we were finding to be associated with longevity could in fact be giving us some important or somewhat relevant information for neurodegenerative diseases, and also for progeria, which is a syndrome of accelerated aging. So the reason we were looking at dementia is because dementia is associated with senescent brain cells, and that can stop functioning well or can also die. Yeah. So we looked at Alzheimer's disease and Parkinson's disease, and so here what I'm showing, we did other analyses as well, but what I'm showing here is the overrepresentation of genes, of the 81 genes, among those that were down-regulated in Alzheimer's, in Parkinson's, and also in progeria. And what we find is that there is a significant overrepresentation of the 81 genes that we found associated with postmitotic cellular longevity, among those which are also have aberrant expression in these two pathologies of neurodegeneration, as well as in the case of accelerated aging. So we also, for Alzheimer's and Parkinson's, we had a higher number of replicas, so we repeated the analysis of co-expression, but this time what we were testing was whether the co-expression of the postmitotic cell longevity associated genes was higher in the controls, and whether the association among those genes associated with cellular longevity were disgregating themselves and no longer working together as a group. And so what we have here is control individuals, and this is the mean co-expression among those 81 genes, and this is what you find in the patients. What we can see is that there is a loss of connectivity or co-expression in those individuals with Alzheimer's, and also we find the same with Parkinson's disease. We all know, well, I think it's very well known that women tend to live longer than men. There is a gap of about 7 years. This is not the same or consistent across different populations or different countries because there are many factors that come into play. So in some countries, men have an even shorter lifespan compared to women because of accidents or war or other factors as well. And in some countries, the gap is also smaller. But overall, in humans, women tend to outlive men by about 7 years. And so what we found was that although there's a lot of dispersion, most of the 81 genes are actually more highly expressed in women compared to men. And this data is taken from the brain, right? So those are the cells in both women and men who have to survive as long as they also do. And this here is data on macaques. So in macaques, the females outlive the males by a much higher proportion of their lives. And what we see is that, and although it's only one additional species, so it may not be a pattern that holds when we do it across many other species, but what we do see is that there in macaques, where females outlive males but an even higher proportion of their lifespan, we find that most of the 81 genes show a higher expression in the females, in the brain of the female macaque compared to the male. So obviously we want to know what kinds of genes are these, right? So we've seen that not only they appear to be working closely together in the most longeved tissues compared to the shorter-lived tissues, but they also seem to be having a signal in diseases associated with a decreased longevity of brain cells, and also in progeria, as well as the comparisons between men and women. So these are the functional categories that come up as significantly associated. Some of them make more sense than others. Cell cycle, cell morphogenesis, and protein folding. So protein folding is a key process to keep postmitotic cells alive, because as you accumulate more errors, you start to produce more aberrant proteins. And so a lot of the protein folding genes need to be dealing with those increasing amounts of errors. This is the full list of those 81 genes. You don't need to remember any of them, and I couldn't tell you any one of them by name either. So I'm just going to conclude and say, well, we did a transcriptomic screen of different tissues with different differing longevities, and we revealed a set of genes that appear to be associated with that. And we found that they also have a series of patterns that are consistent with them having a function in that process. You won't believe me, probably, but we didn't know about this paper, right? It was actually published 2 years ago, and neither ourselves, and I have to say neither the reviewers, noticed that there wasn't a previous paper using a similar approach. So what they did was to use 20 different tissues with known cell longevities. Their approach took tissues that were dated with a different set of techniques. So the one we use is the most accurate, but they also have a larger sample size, so there's a trade-off there. They came up with their own list of, they also have other methodological differences as well, but they came up with a list of about 75 or 55 genes that are positively associated with cell longevity, as well as a list of 155 genes that are negatively associated with cell longevity. And obviously we wanted to test, once we realized there was this paper, whether the lists coincided. And obviously, as you would always expect when you want a good result, you get quite the opposite. So none of the genes actually coincide between the two lists. Having said that, some of the functional categories are enriched on both. For example, cell cycle comes up in both of the lists. And in this paper, they didn't test for any association with disease or male, female, etc. So they didn't do any characterization to confirm the relevance or potential relevance to cellular longevity or aging. But we did that with their list, and it also seems to be consistent, although to a lesser degree than the list that we have. So that's ongoing work. So, and it's been done by Karina, who's also a graduate from, well, no, he's a current student of the undergrad program. So she's working on that. She has actually lots of data, but didn't send me the figures in time. So what I'm gonna switch. Do I have any time left? Lots. Okay, good. I don't work on humans that much, so this is going to be a short talk. So I'm going to change gear and now present some data that has been produced by the people I showed you previously, plus Aline, who's actually repeating everything every time we get additional species being added to the databases. So it's quite fun, and it's a race against time because genomes come up every day. So what she is doing is, rather than comparing different tissues within a single species associated with differing longevity, what she's doing is comparing genomes, and again, not transcriptomes, now it's genomes, of fully sequenced mammals who have differing lifespans. So mammals actually vary enormously in their lifespan. Some species live less than 3 years on average, and others live actually much longer than these, humans and whales as well. So in this analysis, what we were focusing on is whether we can find a signal that is consistently associated with the increase in longevity in mammalian lineages. So what we were focusing on is on gene family expansions and gene family contractions. So gene families can grow when you have instances of gene duplication events, and gene families can contract when you have gene losses. So there are some gene families which are extremely dynamic. For example, olfactory receptor in mammals is one of the largest, if not the largest, gene families. And also they have an amazingly fast gene turnover. So genes are constantly being lost and gained as well. And there is a correlation between how much a species relies on olfaction and how many genes those olfactory receptor families have. There are other instances which are a lot more modest in changes, because with olfactory receptors you can go from 300 genes all the way to 1,600 genes, for example, in the elephant. So there are other gene families that also vary in size, but to a more modest degree. For example, opsin genes. So we know that mammals lost 2 opsin genes during their evolution, maybe because they were hiding from the dinosaurs, and in some lineages, there has been a new duplication of one of the 2 remaining genes that in primates has led to better color vision. So gene families then can be a reflection of the molecular relevance that their members as a group serve within the cell or within an organism. So if the function becomes more relevant, then that can lead to proliferation of the genes in a gene family, and likewise, it can also contract. This is obviously a simplification because there are many ways in which higher functional relevance can be expressed, right? So you can have increases in gene expression or faster molecular evolution, etc. So this is just one aspect of measuring molecular relevance. So before we start, actually, we need to consider the fact that longevity is correlated with lots of other traits. So, for example, we know that larger mammals tend to live longer. So there are exceptions, and those exceptions are actually very interesting, and lots of people are studying how do they live longer than their body mass would predict, etc. But overall, there is a correlation. Longevity also correlates with brain size, and this association holds even when you correct both variables for body mass. So species that have a longer lifespan tend also to have a larger brain. And there is currently a manuscript being prepared that shows which way around that causality goes. So that's something that, not by me, by other colleagues. So we have used these analyses of gene family expansion and gene family contraction to study phenotype of associated with brain morphology. So I'm not going to talk about those, but we basically were using a similar approach. Again, this is another huge table. It's just to show the 51 species that were used in the analysis I'm going to show you, and they spanned a lot of diversity in across the phylogenetic tree in terms of longevity. So this is just to show you how the correlations for the traits that we were controlling for work in the set of species we were looking at. So this is body mass, this is maximum lifespan, and this is encephalization. So encephalization is the size of the brain weighted by body mass. And then what we did was to run correlations between every single one of over 10,000 gene families with or against the phenotype of maximum lifespan across all the species. So for every single gene family, we would run a correlation of the size of the gene family in each species as well against the maximum lifespan recorded for that species. And then we did that and corrected for encephalization, and also we did it for body mass, and we were also correcting for phylogenetic signal. So there are no genes or no gene families that are expanding in concert across all those, in association with all those phenotypes. We found that maximum lifespan is associated with the largest amount of gene families being expanded. That's compared to a relatively smaller amount of gene families associated with encephalization. There is a lot of overlap between encephalization and maximum lifespan, but we found none with body mass. So we corrected only for encephalization and not for body mass. So we wanted to examine what kind of molecular functions these gene families were enriched on. And what we found was that there's a lot of enrichment among those gene families in genes associated with immune response. So the categories we were finding here, using this comparative approach, looking at genomes, were different from those that we were finding looking at transcriptomes within tissues. So there seems to be different mechanisms that we are capturing with this. So this is the A, just shows the categories which are enriched in just with maximum lifespan. This is after correcting for the phylogenetic signal. But actually, when you correct for phylogenetic signal together with correcting for encephalization, then you get immune response being very highly associated. With it, the opposite analysis where we take encephalization as our focal variable, and we don't get 1 minute, and we don't get the same results. So a lot of the signal that we are finding associated with maximum lifespan is not explained by the correlation with encephalization. So what is the relevance to humans, right? Well, even though we were looking at gene family sizes, we weren't looking at mutations or substitutions in amino acids across evolution. And we were looking at all sorts of different mammals spanning hundreds of millions of years in the phylogenetic distance. We still found that when we look at those genes in those gene families associated with maximum lifespan, there is an excess of them that contain SNPs that have been associated with longevity. So there is a crosstalk, we think, between the kinds of genes that are involved in regulating or mediating the evolution of longevity across phylogeny, and to some extent it overlaps with the molecular mechanisms that account for variability within the species in terms of longevity. So what we think is that in order to better understand longevity in our own species, we can have additional or complementary information coming in from comparative analysis. I think that's pretty much it. So the funding has come from those two sources, and moving forward we are working with these wonderful people. This is Dalia del Conde Ovando. She's in Odense, the Max Planck Institute in Odense in Denmark, and she is the director of Species 360, which is the largest repository for demographic data. So we are hoping to be able to look at actual mortality rates rather than the crudest estimates of maximum lifespan. Diego is doing a lot of analysis on sex chromosomes and sex chromosomes decay associated with longevity. And Tamas, he's just a very fun character, and he is working a lot and very interested in the traits that are associated with driving mortality and mortality evolution in wild populations and also across large taxonomic distances. And thank you very much for listening.